Macromolecules. Macromolecules are larger molecules made of smaller parts. There are four main classes of macromolecules. We have carbohydrates, lipids, nucleic acids, and proteins. When we're building a molecule, we put together small units called monomers in order to create a polymer. The polymer is a large unit. Lipids is an exception to this. Lipids have subunits and not monomers. So lipids are not repeating units of monomers. When we're putting together a macromolecule, we do this by dehydration synthesis. Dehydration synthesis is anabolic and endergonic. Anabolic means to build, and one way you can remember that is anabolic steroids is used to build muscle. Anabolic reactions are reactions that put together or build. So we're creating new molecules here. Endergonic means that it requires energy to put into this to get it going. If you take a look at the picture below, we have two monomers here. We're taking an OH off of one and an H off another to join them together, removing water. So we're dehydrating that molecule. Looking at a graph on the reaction, looking at Gibbs free energy, and we'll be talking about Gibbs free energy in a few chapters. When you have an endergonic reaction, it's not going to be happening spontaneously, and you have to put energy in. So Gibbs free energy is greater than 0. When we're taking macromolecules apart, we're going to use hydrolysis. Hydro means water, and lysis means to cut. So we're cutting that molecule with water. If you take a look at the picture here, we're using water, bringing it in, putting an OH back on one molecule and the H back on the other. Hydrolysis is catabolic. Catabolic means to break down, and exergonic, meaning energy is being released. Again, if we take a look at the graph, here, the energy still needs to go in, but not as much to get it going. And Gibbs free energy is actually going to be less than 0. Carbohydrates. Carbohydrates have carbon, hydrogen, and oxygen in a ratio of 1 to 2 to 1. These are our sugars and our starches. They're used for energy or structure. The monomer for a carbohydrate is a monosaccharide. Common monosaccharides are glucose, galactose, and fructose. They're C6H12O6. It just depends on the way that the molecule is arranged and where the functional groups lie as to which sugar it is. Glucose and galactose and fructose are generally found in a ring structure. Disaccharides is when we take two monomers and put them together. Glucose and glucose together are going to create maltose, which is malt sugar. Glucose and fructose, put one of each of those together, it creates sucrose, which is table sugar. And galactose and glucose put together is lactose. An oligosaccharide would be 3 to 10 monomers put together, so a smaller polymer. When we take thousands of these subunits or monomers and put them together, we come up with a polysaccharide. There are 3 main polysaccharides that we can look at. In plants, we have our structure, which is cellulose, and we have starch, which is our energy storage. If you look at the cellulose and the starch, one of the main differences is the way that the molecule is put together. You can notice that this oxygen kind of goes up, and then it goes down, and then it goes up. That creates beta pleats. It's beta linked. In starch or glycogen, you can see that the oxygens are all pointing in the same direction. That creates alpha linkage. That creates more of a spiral. The shape is different because of the way the molecules come together. And remember, shape and function go together. The starch and the glycogen are very similar in structure because they are both energy molecules. Glycogen is energy storage in animals. There is also chitin. Chitin is the structure found in the exoskeletons of bugs and the cell walls of mushrooms. Looking at an evolution link, herbivores eat lots of cellulose, but they don't have the enzymes to break it down. There are 3 different adaptations that have come about in order to help organisms to break down the cellulose. In termites, termites have a symbiotic relationship with a protozoa that lives in its belly that breaks down the cellulose for them. Ruminants will chew their food, the food will go down into the stomach. There are some bacteria there that will help break down the cellulose. They bring it back up, chew it again, and swallow it. So that's chewing the cud. Cows, camels, llamas are all within the ruminant category. Then we have cecotrophs. Cecotrophs create two different fecal pellets. We have the feces, that is the actual poop, and then they create this other pellet that is the cecal pellet. This is partially digested, and what they do is that they will kind of poop it right into their mouth, and they will re-chew that and swallow it again to get more of the nutrients out of there, because they have microorganisms in their gut that will help break down that cellulose a little bit more. It's not a feces, it's a special pellet that's made just for kind of re-chewing their cud, kind of like cows do. Then we have lipids. Lipids have mostly carbon, hydrogen, and oxygen, and remember they are not polymers because they are not repeating monomers. Our lipids are mostly our fats, oils, and waxes, and it's used for long-term storage. Our lipids are nonpolar, and being that they're nonpolar means that they will not dissolve in water. We have triglycerides. Triglycerides is one glycerol with 3 fatty acids attached. We have 2 types of fatty acids. We have saturated, and saturated means that they have the maximum number of hydrogens attached to their carbons in that big long chain. These are generally solid at room temperature. They are things like butter, dairy, fat on meat, and then we have unsaturated fats. Unsaturated fats have a double bond. That double bond creates a kink in the fatty acid tail, and it makes it harder for them to solidify. So these are going to be our oils. They're going to be liquid at room temperature. Trans fats are unsaturated fats. It creates a more dense arrangement of the fat, and these are mostly man-made fats. And then we have phospholipids. Phospholipids have a glycerol with a phosphate group and 2 fatty acids. So one fatty acid is replaced by a phosphate group. These are the main components in your cell membrane. There's a phospholipid bilayer. There's 2 layers of phospholipids. The glycerol head is hydrophilic, and the glycerol tails is hydrophobic. There is water inside the cell and outside of the cell. Those hydrophilic heads are like the water, so they will touch the water, and the hydrophilic tails don't, so they point in. It makes a great shape for the outside of the cell membrane. And then we have steroids. Steroids are fused rings. One main example is cholesterol. Cholesterol is found within our cell membranes, keeping them more fluid. And then we have a number of steroid hormones. Testosterone and estrogen are steroidal hormones. And then we have nucleic acids. Nucleic acids are our information storage, DNA and RNA. The main elements are carbon, hydrogen, oxygen, nitrogen, and phosphorus. The monomer would be a nucleotide. A nucleotide contains of 3 parts. We have a phosphate group, we have a sugar, and then we have the nitrogen base. There are 2 types of sugars. We have ribose, which is used in RNA, and then we have deoxyribose, which is used in DNA. And you can see that the main difference is down here, there's an oxygen. Here, there's just a hydrogen, there's no oxygen attached, so it's without that oxygen. There are 5 nitrogen bases. We have cytosine, thymine, adenine, and guanine. C, A, and G are found in both DNA and RNA. T is found in just DNA, and U is found in just RNA. When our molecules put together, the phosphate group attaches to the sugar of the next nucleotide, and it creates a phosphodiester linkage. That's the type of bond that's formed between the 2 molecules. RNA is a single strand, and DNA is a double strand. When DNA comes together, it actually comes together anti-parallel, meaning one strand goes this way and the other strand goes this way. And then we have protein. Protein is the molecule that does all the rest of the stuff that carbohydrates, lipids, and nucleic acids do not do. Proteins are made out of carbon, hydrogen, oxygen, nitrogen, and sulfur. The monomer for a protein is an amino acid. All amino acids have the same general structure. If we take a look at our monomer, or our amino acid, we have a amine group on this side. On this side, we have our carboxyl, and then we have our R group, all surrounding a central carbon. The R group is where the molecule differs. Depending on what gets plugged in here is going to change the behavior of that amino acid. We have hydrophilic amino acids, and then we have hydrophobic amino acids. This will help dictate the function of that amino acid. Two amino acids are joined together through a peptide bond, a bond between the carbon of the carboxyl group and the nitrogen of the amino group. Here are some of the functions of proteins. This list is not totally inclusive. There are many more functions of proteins. They catalyze reactions acting as enzymes. They could be hormones. They could fight disease, being a part of your immune system. They provide structure. They can be used for transport across a membrane. They can be used for being a receptor on the outside of the cell, or they could be motor proteins moving molecules back and forth. There's a common way that amino acids will come together to form a protein. The primary is simply a chain of amino acids coming together. This is linked together by peptide bonds. So amino acids are linked together through peptide bonds. With the reaction of the carbon-nitrogen peptide bonds, the way that they interact with each other will form the secondary structure. There are no R group interactions here. It's just the carbon-nitrogen backbone that's going to be creating this. And in the secondary can be beta sheeting, which is a back and forth, or we can have alpha helixing, which is this spirally shape. Our tertiary structure is the interactions between R groups. Depending on what R groups are there, if they're nonpolar or polar, will change the structure, and the way that the chemistry happens within that molecule itself will change the shape. And remember, shape and function go together. Quaternary is the kind of optional structure. Oftentimes with proteins, they're very large, and multiple tertiary structures will come together to form a quaternary structure. Changing shape is going to change function. Denaturation is when we break the protein. We can put the protein in different environments, and depending on the environment, it could change the linkages between the R groups and can break the bonds between the R groups, changing the tertiary structure. When we change that tertiary structure, now we're going to change the function of the protein. Things like heat, salinity, pH can all change that, or can all denature a protein, changing that shape, making that protein unfunctional. And this is what you should know.